Let's see if this works. Okay. Thank you all. Let me just set my timer. Thank you all for this. It has been really great. Thank you, Gina, for even the introduction. It's sometimes you don't necessarily feel seen or included, so this was great. So I wanted to talk about eco-evolutionary dynamics of flower microbiomes. And part of that is we know a lot about how microbes affect plants, as we've seen throughout these talks, but we don't necessarily know a lot about what microbes in the flowers are doing, even though flowers are very important for the fitness of flowering plants. We know a tiny bit, as I'll talk about, how they might be affecting pollinator visitation, and there's suggestion that they might be affecting fitness in other ways. So this is kind of how this would all work. We have that there's floral traits that vary, and those might change who is there, what microbes are in the flower, and then different microbes might have different impacts on the flower fitness, and then there might be different degrees of heritability, so some microbes might actually get into the seed and get vertically transmitted. Some might just be associated sort of spatially and temporally, and we don't know how strong that can be. And then we might get a response to selection. So these are all sort of ideas I've been thinking about, but because we know so little, I'm just gonna start with the ecology because we don't even actually know if floral traits act as ecological filters and are selective for particular microbes or not. And so I'm sorry to disappoint, but this might just be an ecodynamics of flower microbiomes talk. And so we don't even really know how flower microbiomes are assembled. Our intuitions are that probably pollinators had a lot to do with it, and there's recent evidence that that might be the case, more or less. But flowers are also exposed to wind and water, and microbes might be coming from all sorts of sources. And so we might have sort of neutral mechanisms of community assembly where things are just kind of landing in the flower, and then we might have sort of drift of some things dying and some things surviving just kind of by chance. Or we might have that some species actually do better, or some genotypes actually do better in certain flowers than others. And so we would have sort of selective mechanisms. And so the little we know about microbiome assembly in flowers, all or almost all comes from yeast in nectar. And what this is really cool work, and the yeast shapes are actually pretty awesome in the nectar. It's just one part of the flower, and it's actually a very low diversity environment in comparison with the rest of the flower. And so different organs in the flower have different sort of environmental conditions, and here is just some micrographs of some parts of the flower, and as you can see, they don't look alike. And I also wanted to pose this to, when we're thinking about microbes, we don't often think how microbes experience the world around them. And so even a petal, which, it does have some textures for us, it's quite a landscape for a microbe. And, like petals might have different sugar exudates and other things, and those vary spatially. And so even within a petal, the microbes might be experiencing many different environments. Pigments change throughout the petal, and pigments have antimicrobial properties. They also have different structural shapes of the cells of the flowers. They change the temperature, the humidity. So this is like highly, highly heterogeneous environment for microbes, and we actually know nothing of how this heterogeneity shapes the microbiome. Furthermore, pollinators also, it's not like they see the flower and they just like grab the whole flower. They have different interactions with different parts of the flower, and different pollinators interact differently with the flowers. So these are all the work I'm going to show is with Mimios guttatus, and these are some of the many different critters that visit Mimios in the field. And so even the same pollinator, so this is just some work I've been doing with another postdoc in the lab, Avery Russell. So this is even bumblebees, depending on what their behavior is, they move different microbes. So we have in the y-axis and the number of microbes they're moving. We mark the microbes with GFP so we can sort of give them microbes in one flower and then let them visit another flower and see, depending on how they're behaving and what part of the flower they visit, they move a different amount of microbes. And so what I want to ask here is, what are the relative contributions of neutral and selective processes in the assembly of Erianthe guttata, that's the new name of Mimios, floral microbiome? And how does the microheterogeneity within the flower contribute to microbial assembly? And what is the effect of pollinators on microbial community assembly? And so to do this work, we went to California to the McLaughlin Nature Reserve, and I sample in 5 different locations because I wanted to get a sort of idea of geographical variation. And in each of those locations, I sample within a serpentine sip. So serpentine sips are great to study this kind of dynamics because the community of plants in the sip is very unique and is very different to the surroundings. So you have sort of these replicate communities. It's also where you can find a ton of Mimios guttatus. And then within those sips, I said, depending on the length of the sip, either 4 or 3 sampling points. And on each of those sampling points, I put a sort of pollinator exclusion treatment. And then at control, I was really worried about humidity and light and other things that the pollinator exclusion might affect in terms of microbial growth. And so I tried to create a control as best as I could that still would allow pollinator visitation. And then carefully in the field, I sterilely, or as sterile as I could, dissected the flower in the stamen, the pistil, and the corolla, and put those in different samples, and then send that to sequence to figure out who was there. And so what we would expect if the organs are acting as sort of selective, if different organs within the flower are acting as selective mechanisms, and different microbial communities will be different depending on the organ. And so if we sort of do this community space, we will see that communities from the same organ cluster together and they are separated from other organs. Instead, if neutral processes are more important, what we would expect is more of a sort of geographical distribution where things are just more similar to whatever is nearby because of dispersal and drift. And so what we would expect is if our sort of community maps are geographical one, more or less. And then we might expect that if we don't have pollinators, there's not that much things coming in, especially if pollinators are the main thing moving microbes. And so then selection within organs might be more important. But if pollinators are moving things all the time and you're getting new things, then you might get that neutral effects are more important. And so what do we see? We do see an effect of organ. And if you have done microbiome, you'll see that those, the variation explained is not terribly bad. But if you do other things, you'll see that there's still a lot that we don't know that's shaping these communities. But organ is the one that explains the most variation of the things we actually measure or control. SIP explains some of the variation, but much less. So that would be a sort of geography proxy. And then the interaction, the pollinator treatment did not explain a lot, and it was not significant, but the organ by pollinator interaction was. And so another expectation we might have if we compare neutral versus selective mechanisms is that sort of the mean relative abundance of a microbe, if you have just neutral, will correspond to the predictive occurrence. So the more abundant the microbe is, the more flowers it will appear in. And so then, if you have something that's way above that, is overrepresented and it's occurring more than you would expect by its relative abundance. And if you have something that's down there, it's underrepresented and somehow is kind of being filtered out. And so what we see is that, in general, we have many OTUs that fit a sort of neutral model, but then we have some that are overrepresented and a few that are underrepresented, especially the petals we get that have actually a lot of sort of, they're the ones that fit the least neutral model, and they have a lot of overrepresented OTUs. And so another expectation we might have here is that if this is all neutral, then as microbes are coming to the flower, they might just be shared between the different organs and they might be all over the place. Versus if they are selective, different microbes might just occur in one or another organ. And what we see here, and I would love to discuss this because it's kind of puzzling, is that the ones that are neutral, the OTUs that fit a neutral distribution in our model are sort of shared among all the organs, as we might expect. But the ones that are overrepresented or underrepresented are actually more shared between organs than what we would expect just by chance. So the ones that are sort of being selected by the flower are also being enriched in all the other organs as well. And so we think that different organs act as ecological filters, but I actually think this might be in a sort of sequential kind of order. So you can imagine the corolla is what's really exposed, is really big, and so microbes get there. That kind of does a first filter, and then from there, things might colonize other organs or not. And as I said before, pollinators interact differently with different parts of the flower, so we wanted to see sort of what actually is the effect of pollinators, because even though we didn't get an overall pollinator effect, we did get an interaction, and that was interesting on its own, especially because we did have the hypothesis that if we excluded pollinators, we might see a strong filtering effect, versus where in our control, where we have pollinators, we expected a more sort of neutral effect. And that's kind of what we see. So on the x-axis are different beta diversity indices. So this just kind of happens across all the indices we use. And then we separate, where we did our analysis, this time separating the no pollinators and our control treatment. And we see what's the percentage variation explained by organ dependent. And as you can see, when we don't have pollinators, organ explains almost double of the variation as when we let them in. And so we do think that they are having a sort of swamping effect where they're dispersing microbes and kind of erasing some of the selective mechanisms. Moreover, pollinator visitation is not like you exclude them and that's it. You also have variation in how many in the rate of visitation in different locations. And so we wanted to see sort of, if we look at the difference in composition of our exclusion treatment and the treatment we allow pollinators to visit, we can have a sort of distance measure, and that's going to be sort of the effect of pollinators on community structure. And we could do that for all of our locations that might vary in pollinator visitation. And so what we would expect is that if we don't have a lot of pollination, we won't see a lot of difference of our treatments because they'll basically be no pollinators and almost no pollinators. Versus if we have a lot of pollinators, then that difference should be larger. And so what we see is that when we look at the visitors that are sort of external, so butterflies and things that might just like touch the corolla but not really go in the flower, we see a relationship in the petals between visitation rate and this sort of pairwise beta diversity, this pollinator effect measure. But when we look at things that go in the flower, we see a relationship with petals that's even stronger, and we actually start seeing a relationship with the stamen. So this relationship is high in magnitude, is not significant because, as you can see, the variation is kind of crazy, and we would like to have more samples for this. Another interesting part here is, I don't know if you can see those blue dots, the style, blue squares. There's no relationship whatsoever between pollination visitation rate and the difference in. And so what we think we have is a sort of dispersal selection balance shaping the microbial communities in the flowers, where when we have a lot of pollinators, we have these sort of mass effects. All of it is shaped by pollinators, versus when we have less pollinators, we might have a stronger effect of sort of species sorting within the flower, where different organs have a different microbial community. And what's cool here is that then we can make predictions about how disturbances might affect microbial communities in flowers. And so we would expect that things like fire, fertilizer, pesticides that will affect the pollination and also a flower will shape mostly the dispersal part, whereas things like fertilizer, warming, drought might affect the sort of changing the floral chemistry and the floral microenvironment, and that might shape the selection mechanisms. And so now we're starting to look at actual floral traits, so we are looking at color, number of trichomes in the petals, and all sorts of array of traits, and see how they shape microbial community. And hopefully we'll start measuring the effects on fitness, having high loads or low loads of microbes in the flower. And so with that, I would want to thank you all. And then I would want to thank the Ashman Lab, Rebecca Hayes, who, an undergrad at the time, which she helped a lot with the pollinator observations, and now she's helping me develop a curriculum using the plant trait microbiome work. And then a lot of other people that have gotten really good feedback on this. So thank you.